is Caitlin White, and I'm going to talk to you today about lab technique CBC versus CBC with manual dip. The purpose of this lab is to distinguish between machine-operated CBC and a manual CBC, which is a lab technician actually counting them himself and getting a more accurate result. Also, to explain why a physician would order this test or any healthcare provider over just a automated CBC. And then also to explain the procedure, the actual lab technique of how to do this. How to collect this specimen is very important in this process. As you can see here, there's multiple different tubes that you can choose to collect the blood from a venipuncture. Some of these they all have different substances in them, coags, sodium heparin, and things like that. It's very critical that you collect the blood from the lavender tube, and also you can do it out of this microtainer as well. The lavender tube has EDTA in it, and that keeps the blood from clotting, and it makes for more accurate results. After that, after you collect the blood, you're going to invert it a couple of times. It's really important that you don't shake the tube or just leave it sitting there. Shaking the tube could lead to hemolysis, which is the breakdown of the cells, and that could lead to skewed results. This is how to actually count the white blood cells and red blood cells in your field of view. So you're going to collect the blood, and then you're going to proceed to set up your slide. You're going to take a pipette and draw out a couple mils, and then put 2 drops of blood onto your slide, and then you're going to put a lab slip or a cover slip on top. Whenever you do this, just put it underneath the microscope, get your microscope in the field of view, and then make sure there's a clear distinction between the red blood cells and the white blood cells. The red blood cells will have a hollow center and kind of like a donut, and the white blood cells will be a little bit darker. Keep in mind when I talk about white blood cells, I'm talking about leukocytes, and that is the 5 different kinds of infection-fighting cells in your body. So they're not going to be all the same size or the same color, but they will be a little bit darker than the red blood cells. So you have your big field of view, and then what you're going to do is you're going to divide your field of view up into 8 sections, or whatever you feel comfortable manually counting. So I would divide it into 8 sections, and then manually count every single white blood cell and every single red blood cell. And then you're going to multiply that number, so mine would be 8, and then that'll give you the total number of that in the little microscope field of view. This formula below is to calculate the ANC, which is the absolute neutrophil count, and that's the measure of infection-fighting diseases, or infection-fighting white blood cells in your body, or in that sample of blood. If the ANC is lower than 1,000, you're at a very high risk of infection, and every time that just decreases, your risk for infection increases. The normal ANC is about 2,500 to 6,000, and too many white blood cells indicate a disease or disorder. Same with not having enough white blood cells to fight off infection. If this is found in the blood, you should run more tests to accommodate the findings. And then this right here is a picture of what a picture underneath the microscope might look like. This is of a healthy person. They don't have many white blood cells. And then this is how you would collect the blood or place it on your slide. It's just a pipette and a purple top, and just extracting a couple of mils. And then some of y'all might be wondering why you would want to do this over a automated CBC, or why physicians would want to order this test. The purpose of ordering a manual CBC is just to get a more accurate reading. Just with people, machines have errors too, so if one is a little skewed, you could ask for a manual CBC and get a more accurate reading. This is the machine, it's called the LH machine, that's in the lab. It's a lot bigger in the lab, but this is the one that automatically runs the desired test. But like I said, there's error with machines and technology, just like there are with humans. So this is how to manually count a CBC, and I hope you learned something. Thanks.